Well, I'm Dr. Barney Reese from Biological Sciences, and I've been asked to come today to talk to you about what I do in my research lab and how that might relate to what you guys are going to be doing today in lab. So, in general, I'm interested in the effects of low oxygen on animals and in fish in particular. And we all know that oxygen is important. We have a high requirement for oxygen because that's basically the way that we generate most of our energy through aerobic metabolism. So because of that, if we face conditions of low oxygen, we don't do very well. So you're probably familiar with the fact that if you are deprived of oxygen for more than a few minutes, then you can suffer some pretty serious consequences. Brain damage, heart damage, and you're not going to be around very long in the absence of oxygen. And when we think about the aquatic environment, though, there are many instances when the oxygen is low in the aquatic environment because of a variety of factors. Warm water, stagnant water, high salinities. All these things can contribute to a decrease in the oxygen availability in the water, and yet the fish and the animals and plants that live there have to deal with this. So I'm interested in how fish in particular deal with conditions of low oxygen. And because one way that we generate energy is through aerobic metabolism, when oxygen goes down in the environment, then the capacity for energy production via that process is going to be reduced. So they have to come up with alternative strategies to generate energy, or again, they're going to face some pretty nasty consequences. So one way to do that is to increase the amount of energy that's produced by anaerobic glycolysis. So you guys have all heard about anaerobic glycolysis, I suspect, in your freshman biology or even high school biology classes, right? No? Well, it is a way to burn carbohydrate that allows the animal or the organ or the tissue to generate a small amount of energy, even though they cannot burn it completely because oxygen's not available. So you can think about glycolysis as the partial metabolism of carbohydrate, and it's useful because it allows some energy formation, but it's not the same amount that you might get from a completely aerobic processing like when oxygen is available. Nevertheless, it does represent one mechanism for energy acquisition in the case of low environmental oxygen. And so one way in which fish might respond is by an increase in this process, this anaerobic metabolism, and the product of that anaerobic metabolism is lactate. And you guys probably are familiar with lactate because if you've ever exercised, which I think most of you do exercise, if you exercise strenuously, then you have an accumulation of lactate in your muscles for the same reason. Your muscles are relying upon the partial breakdown of carbohydrate, and the product of that partial breakdown is lactate or lactic acid. So if you've sprinted or worked out vigorously for short periods of time, then you might get that burning sensation in your muscles, and people attribute that to the accumulation of this end product of that anaerobic metabolism. Well, the enzyme that's involved in that step which produces lactic acid is an enzyme which I think you guys are going to measure today in lab, and that's lactate dehydrogenase. So all you have to know about lactate dehydrogenase is that it's involved in that process where you can get some energy from the incomplete breakdown of carbohydrate, and it is the last step. So by measuring the activity of lactate dehydrogenase, then you get an idea as far as the tissue capacity for that process. And again, some animals are better than others, so fish are better than rats. But even within your body, some tissues are better than others. And I go back to that example of exercise. When you exercise, you have some muscles that are better at the anaerobic formation of energy. They have more capacity for lactate formation, and they have more of that enzyme, lactate dehydrogenase, than other muscles. And you're familiar with this if you've ever eaten chicken, because you know the difference between white meat and dark meat. And white meat is that meat which has a high capacity for anaerobic metabolism, and it has a high capacity for lactate formation. So you would expect that it would have a high amount of this enzyme, lactate dehydrogenase. Whereas red meat is red because it has myoglobin, it has blood vessels, and it has a greater capacity for aerobic energy formation and low capacity for anaerobic energy production. And so it's going to have a low capacity for lactate formation, and it ought to have a low capacity for, or a low amount of the enzyme lactate dehydrogenase. So, in a nutshell, I'm interested in fish and how they respond to low oxygen, and I can understand a little bit more about when they'll do well and when they'll do poorly when the oxygen tensions or the oxygen levels in water change, for example, due to human influences. But the same process that I'm studying in fish has bearing on humans and mammals and vertebrates in general, because the same processes that fish use to do better during conditions of low oxygen are the same features that allow your muscles to perform vigorous exercise for those short burst types of activities. That is, the high capacity for this alternative mechanism of energy production, which does not require oxygen, but it does involve the enzyme lactate dehydrogenase. So these are also issues that you ought to think about, because the occurrence of low oxygen is part of a variety of diseases that are important in human health, namely stroke, heart attack, cancer. All of these things are associated with oxygen deprivation to specific tissues, the brain, the heart, or other tissues. And those tissues that tend to survive better are those that have these alternate pathways of energy production. And this is, in fact, one reason why cancers tend to do better than your normal body cells, because they have a higher capacity for anaerobic metabolism. They can withstand conditions that your normal body tissues would not be able to withstand, thereby allowing them to grow and proliferate. So although we study fish in the context of environmental low oxygen, what's happening out there in the swamp, what we hope to find in the lab should have its counterpart in human health and disease. And hopefully you guys will get a lot of great data on lactate dehydrogenase from mussels, and show that there are different mussel phenotypes that are related to this capacity for anaerobic metabolism. Cut.
Thanks a lot.
All right. And if anybody wants to talk more about what I do in the lab,